Good morning, everyone. Welcome back to my channel, Molecular Mysteries. Hope everyone is doing fine out there. So, without any further delay, let's get into the class.
Come and fly away with me. Come and fly away with me.
Good morning, everyone. Hope everyone is following the class till now. Okay, so in the last class, we have discovered about the cell biology, about the important cycles, all important cell divisions and their significance, right? Even the chromosomal structure is also discussed, right? So the shapes of chromosomes and all. So from here after I want to discuss the basics of the molecular genetics, so because it is one of the important topics given in your syllabus pattern, so I hope this would be useful for the undergraduate students also. Okay, so please go through it. So this class I want to talk about the molecular genetics. What does molecular genetic mean? Like, it's at the genetic level in molecules. So how the genes are decided, and how the character of a person, like it's a phenotype, is decided. So this class is all about the basic terminology that you come across the genetics, because hereafter I keep using the same words, so you should be aware of the exact meaning of that. Okay, so before that, what is a gene? So each and every person has got genes on the chromosomes. So if you go back to the structure of chromosomes, you have two sister chromatids present per one chromosome, right? So each and every a person is mainly generalized or mainly judged by the gene. Each and every character has got a gene. It could be the height, it could be the color of the color of the skin, or it could be color of your eyeball, like that. So each and every character of a person is mainly decided by a gene. So gene is considered to be the basic unit of your inheritance. Okay, so genes are passed from parents to offspring and contain the information needed to specify the physical and biological traits. So roughly, even per cell, if you take in humans, you have around 30,000 to 40,000 genes present. That's a rough figure. Okay, so you can imagine how many genes are really, are judging our character, or judging our physical appearance. Okay, okay, in this class I want to talk about that. Okay, so genes come in pairs in the same way of chromosome. This is important. As I told you, genes generally come in pairs. Okay, each parent of a human being carries two copies of their genes, one coming from the parent side and one coming from your mother side, or and paternal side. Okay, and the whole genetics, you can say that Mendel is considered to be the father of genetics, Gregor John Mendel, because Mendel's laws and all. Maybe you should have gone through that in your plus 1 and plus 2, like 11th and 12th classes. You could come across it. So I just want to brief everything in this first class of your molecular genetics. So before I enter into the main part of your genetics, I just want to have you a clear definitions of all these basic terms. So here you have already gone through the phenotype, genotype, everything in the last class itself, right? So this time I want to talk about what are these terms. So character, I told you each and every person has got a character, right? Each and every character is mainly decided by a gene, and gene is present on the surface of your chromosome, like what you call as gene locus. See, gene locus is where your gene is approximately present on the surface of your chromosome. So if you go back to the previous class where you told that you have two sister chromatids for one chromosome, each and every character is also having a contrasting trait. Like, if you take a height of a human being, height of a person, tallness and dwarf are the two contrasting traits. I can tell you they are called the contrasting traits of one character. So for each and every character you choose, there are two contrasting traits, and those are called the alleles. See, here you have come across the term alleles. So alleles are the two alternative forms of a gene, right? Like, if I take the color of a person, or if I take so what has Mendel done is, it was all studied in Pisum sativum. It's the name of the plant, the biological name of your piece. Okay, Pisum sativum. There he has taken 7 different characters. So each and every character has got two different contrasting traits or alleles, so based on which you can decide the color of your pot, color of your cotyledon, and height of your stem, and height of your leaf, everything. Okay, and also the shape of your seed, whether it is a completely wrinkled seed or it's a good round seed. Okay, everything is decided based on the character, which is mainly judged by the gene. So that's why gene locus, I told you, it's a place where genes are present on chromosome. Alleles are the alternative forms of a gene. And homozygous and heterozygous, here don't get confused. So for that I may need to draw the diagram. I will. This is your chromosome, one cystochromatid, and this is your another cystochromatid. These both are cystochromatids. If you take the characters male, I am taking the height of a person. Okay, this height of a person you got is T, and this is T. See, the height of the person is a gene. It is having two alleles, which is tallness decided by capital T, and dwarf, which is a small t. Right? See, this if this is the condition, this is what you call heterozygous. Sorry. See, this is one. This is coming from your maternal. For example, this is the example that I'm giving you for understanding. This is your parental. So these are the genes related to the height of the person, and this is your tallness gene, and this is your dwarf gene. So this condition is called the heterozygous. Okay, and in the same way, if you go for if the both alleles of the sister chromatids are same, they are called homozygous. See, here comes the important term, what is dominance and what is recessive. So which is expressed more is what you call dominance. The one which is expressed little is called recessive. For example, so what happens is, so one you got to know, this is your homozygous. This is also homozygous because you have two copies, right? But this is your heterozygous. Okay, these three are the genotypic expression, right? Expression. If you go for the phenotype, if I'm talking about the phenotype, this is tall. If I'm talking about phenotype, these people are tall because they have two copies of your dominant gene, and these people are dwarf. Phenotypically they are dwarf because they have two copies of the recessive gene, and these are T and T, these are the heterozygous, but this is the dominant one in heterozygous part. That is what I want to talk to you. Both they have one copy of tall gene and one copy of dwarf gene, but only one is getting expressed in phenotypically. So these people are tall, though they are heterozygous. Okay, see, if you don't get this phenotype is a physical appearance, and genotype is the genetic expression or genetic makeup of an individual. Okay, and if you say this is this here, though it's a two copies are present, this is expressed more. Okay, and hence the people with the heterozygous condition still are tall. Okay, if you go for this condition, this is homozygous again because you have two copies of dwarf gene, so people are dwarf. This says that the recessive character would only be expressed in homozygous condition. Please remember, the recessive gene can be phenotypically expressed only when it is in homozygous condition. When it is heterozygous condition, the recessive is cannot be expressed. Only the dominant gene is expressed. Okay, so this is how you generalize it, like how you really read a character. You have to get to know the contrasting traits of every gene, then only you will be able to draw these homozygous and heterozygous conditions. So hope I am clear with this all. Please let me know in the comment box if you have any doubts regarding this, because these are the basic terms that you have to get to know the basic terms before you enter into the main subject of your molecular genetics. Okay, so these are the basic terms. Okay, so I think I got a better idea. So if you go back, I told you gene locus, I have told you alleles, I have tell you, sorry, dominant and recessive, which is expressed more as dominant, and the recessive can only be expressed in homozygous condition. Right, in heterozygous condition, dominant will only be expressing. So phenotype, genotype, haploid, diploid, and homologous chromosomes. Right. So monohybrid and dihybrid crosses. So for this, what they have done is, what is monohybrid cross? So these are the Mendel's laws. Okay, Mendel's, what he has done is, these questions may be asked, I just want to cover it for your better understanding. Okay, so what he has done, he has taken the height of a stem, like, which is a pisum sativum. Told you it's a biological scientific name for your piece. Okay, so if you take the height of the stem, please excuse me with the sound. This is the height of the stem, and what he has done is, he has taken the Mendelian character. So each and every height of the stem is having two different contrasting traits. Okay, and that is tall and dwarf, I told you, right? So he has crossed between them. So like, the first cross is, first this is the tall gene that he has chosen, and this is the dwarf plant he has chosen, and it is expressed only in the homozygous condition. Right, when you cross between them, what happens is, it gives only one type of gamete, it gives only one type of, sorry, so hence producing this one. So this is your P1 generation, and this is your F1 generation. Now he has self crossed again with the same heterozygous. Now imagine you get two different gametes. Right, right. So this becomes, so these are tall, these are heterozygous, but still they are tall because this is dominant and these are tall, but this is the only one which is dwarf. So if you go for the phenotypic ratio, in monohybrid cross, they may ask you, phenotype is a physical appearance. So if you say the physical appearance, the ratio is 3:1, because only you got 3 tall and 1 dwarf, right? So instead, if you go for the genotypic expression, so hope I am clear. So this is the monohybrid cross, where he has taken only one character, and what character is the length of the Pisum sativum plant. Okay, so within the one class itself, he has divided the phenotypic expression and the genotypic expression. In the same way, he has done the dihybrid cross also. So there he has taken two different characters. If you want, I can give you again, I can draw for you. If you want, I can do it, because it's a time taking process. I will attach the same important thing in the dihybrid cross also. So for the dihybrid cross, you have to two different characters are taken, for which you have two different contrasting traits, right? So it's a big process, and again you have to make the ratio and you have to cross it. That is how the he has presented the dihybrid crosses and monohybrid crosses. Okay, so that's what. So here I told you, you have two generation, the genotypic expression and generation phenotypic expression, right? The same way what he has done, he has gone for the dihybrid cross. So he told the cross involving two pairs of purulent contrasting traits is called dihybrid cross. I told you he has taken the color of the pod and the shape of the seed. In the monohybrid cross, he has taken the length of the stem and he has taken the color of the pod and the shape of your seed. So they have been crossed, and when you go for the F2 generation genotypic and genophenotypic ratio, these are the things that happen. Okay, so if you want, I can draw it. If you please let me know in the comment box. So what are the still modifications that he has done for the monohybrid cross? That is incomplete dominance and codominance. What does that mean? See, for example, if you take an example of snapdragon, it's the name of a plant. Okay, so what he has done, he has crossed the red color flowers of snapdragon with the white color flowers of snapdragon. So based on it, what should happen? You should come to know either the red should be covered in the progeny or the white should be covered, like, because being a dominant, which one is a dominant we don't know. So based on the dominant gene, the progeny will also be getting, the F1 generation will also be getting either the red or the white. But what happened, the progeny ended with the pink flowers, which says that there is no complete domination, there is no complete expression of your the red flowers. At the same time, there is no complete expression of your white flowers. It's a incomplete domination. There is no complete the phenotypic expression of the red is completely suppressed. At the same time, the phenotypic expression of the white is also suppressed. Does mean they're having the partial domination, not complete domination. So they're called incomplete domination. Hence, the F1 generation is ending up with pink flowers. So that is your example for incomplete dominance. And codominance, yes, this is very important because blood grouping is something you should get to know why, what are positive, what is positive, what are positive blood groups, and how the cross happens, because you can even decide the progeny with the parents blood group. In the next class I will also detailedly explain about showing the pictures and also. Okay, so till now, why it is called codominance? Because see, these blood groups, for a blood group, for a character called blood group, you should have two important contrasting traits, but you have multiple alleles, like not instead of having only two alleles for a gene. These blood groups are decided by many alleles. They are called the example for multiple alleles. So blood groups are the examples for the multiple alleles, where you have A group, you have B group, you have O group, and you have AB group also. So each and the blood character is decided by four different alleles. So in the next class I will talk about the blood grouping and why is it called antibody and antigen, everything. Okay, so I hope I'm clear with the basic terms in molecular genetics. In the coming class I will talk about the blood groups, and then in the next classes also I will also discuss about the dominant X-linked characters, Y-linked characters, XY-linked genes and all. Okay, so this may take 1 or 2 weeks for the molecular genetics to cover. Okay, so thank you and have a nice day. So everything will be linked in your uh, description box. Please go through it, and if you have any if you have any doubt, please let me know in the comment box. So hope I am clear with the basic terms. So thank you. See you in the next class. Until then, stay positive, stay healthy, and be kind to everyone. Thank you. Bye bye.